this presentation, we will review the structure and function of the kidneys, ureters, and bladder, which constitute the urinary system. The paired kidneys produce urine that is transported by the ureters to the bladder. Here it is stored until it is excreted through the urethra to the exterior of the body. The kidneys play a major role in maintaining the extracellular fluid composition and volume by repeatedly filtering the plasma, returning back to the blood necessary elements, and eliminating unnecessary or unwanted elements in the urine. Using our specimen, let's examine the relationships of the kidneys, ureters, and bladder to other abdominal structures. The anterior abdominal wall and some of the abdominal viscera, as well as the parietal peritoneum, have been removed so that we can see the location of the urinary organs more clearly. Because the kidneys lie between the parietal layer of the peritoneum and the posterior body wall, the kidneys are said to be located retroperitoneally in position. Notice that the right kidney is slightly lower than the left kidney because the liver occupies more of the abdominal cavity on the right side of the body. Let's take a closer look at the left kidney. A tough, protective renal capsule encloses the kidney. The renal fascia and adipose capsule that surrounds each kidney has been removed. Perched on top of each kidney is an adrenal gland, so named because of its anatomical location. Adrenal means next to the kidney. The adrenal gland secretes a host of hormones with diverse functions, many of which are unrelated to the urinary system. In the center of the kidney's concave side is a notch called the renal hilus. Entering and exiting the renal hilus are the renal vein and the renal artery. Blood processed by the kidney passes through these vessels. Notice that the length of the renal arteries and veins on the left and right side of the body are different. Since the inferior vena cava lies to the right of the midline of the body, the right renal vein is shorter than the left renal vein. Conversely, the aorta lies more on the left side of the body, so that the left renal artery is shorter than the right renal artery, which lies posterior to the inferior vena cava. The other large structures located in the hilus regions of the kidneys are the ureters. The ureters drain the kidneys of newly formed urine. Smooth muscle in their walls propels the urine towards the urinary bladder. As the ureters descend through the abdomen, they cross the common iliac blood vessels and enter the pelvis. They deliver urine to the urinary bladder through two openings on its posterior side. The bladder is located in the pelvic cavity behind the pubic symphysis, which helps to protect it from injury. Using a sheep kidney, which has been sectioned in a coronal plane, we can review its internal anatomy. The outer portion of the kidney is the renal cortex, which surrounds the inner renal medulla. Invaginations of the cortex into the medulla are the renal columns. These columns separate the medulla into several cone-shaped structures called pyramids. Notice the orientation of a pyramid. Its base abuts the cortex, and its apex is located deep in the medulla of the kidney. The pyramids contain microscopic nephrons, which are the sites of urine formation. Each kidney contains approximately 1 million nephrons within its pyramids. The nephron is a hollow, fluid-filled structure which functions to filter and process the blood. Although the nephron is a continuous structure, it is arbitrarily divided into segments based on its structure and function. The first part of the nephron is the glomerular or Bowman's capsule. It is a cup-like structure that surrounds a knot of capillaries referred to as the glomerular capillaries, or more simply, the glomerulus. The glomerular capsule and the glomerulus constitute the renal corpuscle, which is the site of filtrate formation. The cells of the capsule and capillary are modified so that the plasma filters easily into the capsule. The outer or parietal layer of the glomerular capsule is formed by a continuous layer of simple squamous epithelium. The inner or visceral layer lies adjacent to the glomerulus within the capsule. This visceral layer is modified to allow filtration of the blood into the capsular space and is mostly made up of cells called podocytes. The podocytes have many foot-like structures called pedicels, which surround the glomerulus and share its basement membrane. The spaces between pedicels are called filtration slits. Extending between these filtration slits are slit membranes. Together, these slit membranes and the shared basement membrane regulate which materials, based on their size and charge, are filtered from the blood into the capsular space. The capillary endothelium is also modified. Pores in the endothelium of the capillary, called fenestrations, increase the permeability of the capillary. Except for most proteins and blood cells, the fenestrations allow everything in the plasma to be freely filtered. Beyond the cup-like glomerular capsule, the nephron changes to become tube-like. The initial portion of this tube twists and turns to form the proximal convoluted tubule. A single layer of cuboidal epithelium forms the wall of the tubule. The luminal surfaces of these cells bear microvilli, which facilitate reabsorption and secretion by increasing the surface area of the plasma membrane. Large mitochondria are also present in the epithelial cells of the proximal convoluted tubule. The next portion of the nephron contains both thick and thin tubular portions leading into and away from the curved loop of Henle. The loop of Henle is essential in the formation of concentrated urine. The epithelium forming the wall of the thick portion of the descending limb of the loop of Henle is continuous with that of the proximal convoluted tubule. As the tube thins, the epithelium changes into a layer of simple squamous epithelium, which is freely permeable to water. The simple squamous epithelium continues around the loop and into the ascending loop of Henle, where it begins to increase in height, becoming cuboidal to low columnar epithelium. This taller, thicker epithelial layer forms the walls of the thick ascending limb of the loop of Henle. This portion of the loop of Henle is not permeable to water, but does contain sodium chloride pumps. From the ascending limb, the filtrate moves into a second twisted segment called the distal convoluted tubule. Simple cuboidal epithelium forms the wall of this portion of the tubule, as well as the wall of the collecting duct. Each collecting duct receives urine from several distal convoluted tubules. There are two main types of nephrons in the kidney. Cortical nephrons are the most numerous and reside mostly in the cortical region of the kidney. Only a small portion of their relatively short loops of Henle enters the medullary region of the kidney. In contrast, juxtamedullary nephrons lie close to the border between the cortex and the medulla of the kidney. Their long loops of Henle dip deep into the inner medullary regions. The glomerular capsules of both types of nephrons lie in the cortical region and give it a granular appearance. One difference between the two types of nephrons is the length of their respective loops of Henle. The long loops of the juxtamedullary nephrons are essential to the process of concentrating urine. After the urine is formed in the nephrons and is collected in the collecting ducts, the collecting ducts unite and form larger papillary ducts, which drain toward the apical end of a pyramid, a region called the renal papillae. A minor calyx surrounds the papillae and collects urine as it drains from each pyramid. 2 or 3 minor calyces join together to form a major calyx. Newly formed urine collected in the major calyces empties into the expanded renal pelvis, which continues at the hilus to become the ureter. Both the major and minor calyces are extensions of the renal pelvis. The kidneys are blood processing organs regulating blood volume and composition. As such, they receive a relatively large proportion of the blood flow. At rest, the renal arteries deliver about 1/4 of the total systemic cardiac output of blood to the kidneys, which is about 1,200 milliliters per minute. Each renal artery enters the hilus of the kidney and divides into several segmental arteries. Each segmental artery further divides to give rise to the lobar arteries, which extend through the pelvis and branch into interlobar arteries. The interlobar arteries course between adjacent pyramids in the renal columns to terminate by branching at the base of the pyramids. The branches arch over the pyramids as the arcuate arteries. These arcuate arteries give rise to the interlobular arteries that extend into the cortex. In this illustration of a nephron and its microvasculature, we can follow the flow of blood from the interlobular artery. Each interlobular artery furnishes blood to several afferent arterioles, each of which continues to become the glomerular capillary network surrounded by the glomerular capsule. Leading away from each glomerulus is an efferent arteriole. Each arteriole gives rise to a second capillary bed. The paratubular capillaries, which surround the tubules in the cortical region, and the vasa recta, which surround the ascending and descending limbs and loop of Henle in the renal medulla. These capillary networks empty into interlobular veins. Blood flows out of the kidney through veins, which accompany the arteries of the kidney. Blood from the interlobular veins empties into the arcuate veins, which return blood to the interlobar veins. These veins empty into the renal veins, which then drain into the inferior vena cava. In order for the kidneys to function properly, blood pressure and the rate of filtrate formation must be regulated. These activities are controlled in part by the juxtaglomerular apparatus. It is located in the region where the distal convoluted tubule passes through the fork formed by the afferent and efferent arterioles. The tubular and vascular cells are specialized in this area. The specialized tubular cells form the macula densa. Feedback from the macula densa to the arterioles provides signals to regulate the filtration rate. The afferent and efferent arterioles contain juxtaglomerular cells, which are modified vascular cells sensitive to macula densa signaling. In addition to responding to signaling from the macula densa and altering blood flowing through the glomerular capillaries, they contain large secretory granules containing renin. Renin is an enzyme that sets off a pathway involved in the long-term regulation of blood pressure. Let's take a quick look at the renal processes that occur in the nephron which result in urine formation. About 20% of the plasma entering the glomerular capillaries will leave the circulation and be filtered into the glomerular capsule. This process is non-selective. Everything in the blood is filtered except for blood cells and most plasma proteins. The plasma that enters the glomerular capsule is now referred to as filtrate. About 99% of the filtrate is returned to the blood in the peritubular capillaries or vasa recta. Since this process involves putting something back from where it came, it is called reabsorption. This mechanism allows the body to keep necessary nutrients such as water, glucose, amino acids, and ions including sodium, potassium, calcium, and chloride. Secretion occurs in the opposite direction of reabsorption. Secretion is the movement of substances from the blood into the filtrate. This process allows the body to get rid of substances such as hydrogen ions, ammonia, and drugs at a faster rate. In summary, as the filtrate travels along the nephron, necessary elements are returned to the blood and unwanted substances are added to the filtrate. Whatever remains at the end of this processing enters the collecting duct and is now referred to as urine. As shown in our specimen, the renal pelvis from each kidney is drained by a ureter, which transports urine to the bladder. This histologic cross-section of a ureter shows that its wall is composed of 3 layers. The innermost mucosa layer lines the lumen and contains transitional epithelium resting on a lamina propria membrane. This type of epithelium is specialized for changing shape and stretching when the ureter fills with urine. The middle muscularis layer contains an inner longitudinal layer and an outer circular layer of smooth muscle. Alternating contractions of the smooth muscle in the muscularis results in the action of peristalsis, which moves urine along the ureter toward the bladder. The superficial layer of tissue, which forms the walls of the ureters, is the adventitia. This areolar connective tissue layer contains blood vessels, lymphatics, and sympathetic and parasympathetic nerves that supply the mucosa and muscularis layers. We will use a male hemisected specimen and illustrations to review the internal anatomy of the urinary bladder. The bladder lies posterior to the pubic symphysis and anterior to the rectum. Lying inferior to the bladder is the prostate gland. The urinary bladder is a hollow, muscular storage organ. When empty, it has a pyramidal shape and its internal wall is thrown into folds known as rugae. As it fills with urine, the bladder extends superiorly toward the abdominal cavity and its rugae flatten out. The floor of the urinary bladder is smooth and contains a triangular-shaped area called the trigone. We will examine the trigone in this illustration of an anterior view of the bladder. The trigone, a small inverted triangle, is defined by 3 openings. The upper 2 openings are the locations where the ureters open into the bladder, and the apex of the inverted triangle is the opening from the bladder into the urethra. Micturition occurs when contraction of the bladder expels the stored urine through the single urethra. The histology of the bladder wall is similar to the distal end of the ureter. The inner mucosal layer contains transitional epithelium resting on a lamina propria membrane. The inner longitudinal, middle circular, and outer longitudinal layers of smooth muscle form the muscularis layer. These 3 layers of smooth muscle are collectively called the detrusor muscle. The urethra conducts urine from the bladder to the outside of the body. 2 sphincters control the excretion of urine from the body. One of these, the involuntary internal urethral sphincter, is a thickening of the circular smooth muscle layer of the detrusor muscle located near the junction of the bladder and the urethra. The second sphincter is the external urethral sphincter and is voluntarily controlled. It's composed of the skeletal muscle fibers forming the urogenital diaphragm, which encircle the urethra as it passes through it. The urethra conducts urine and semen. It travels through several structures to reach the exterior and contains three named portions. Lying inferior to the bladder is the prostate gland. The proximal prostatic urethra travels through the prostate gland, an accessory gland of the male reproductive system. The middle part continues through the urogenital diaphragm as the membranous urethra. The distal portion travels the length of the penis as the spongy or penile urethra. It opens to the exterior at the external urethral orifice. In our female specimen, we will now identify the location of the urethra and some of the other structures of the urinary system. The urinary bladder lies behind the pubic symphysis and in front of the uterus. Anterior to the vagina is the urethra. The urethra in the female is a short tube, less than 4 centimeters long. It opens to the exterior at the external urethral orifice. This concludes our presentation of the urinary system.